Good morning everyone, I am Cheri Domingo, your pre-service teacher. So today we're going to discuss about Chapter 3 of cell structure. Before we start, let us know first the objectives for this lesson. First is to identify the cell structure, explain the difference between prokaryotes and eukaryotes, and assimilate the importance of learning the cell structure. The topic to be discussed is cell, metabolism, DNA, and there are two categories of cell, which is eukaryotic and prokaryotic. Some microbes are prokaryotes, which is bacteria and archaea. Some prokaryotes are algae, protozoa, and fungi. Some are not composed of cell, which is virus, prions, and viroids. This chart represents in how microbes can be classified into two, which is acellular and cellular. In cellular, there are two types of the cellular, which is prokaryotes and eukaryotes. Example of a cellular are viroids. These are infectious RNA molecules. Prions are small infectious proteins that apparently cause neurologic disease in animals. Viruses, it is any of numerous kinds of very simple organisms smaller than bacteria, mainly of nucleic acid in protein coat. Since we are talking about cells, we will now focusing on cellular. Cellular, it is relating or consisting of living cell. Cellular can be divided into two types, which are prokaryotes and eukaryotes. Prokaryotes is an organism that has less complex cellular structure, while eukaryotes, organism that contains more complex cells containing a true nucleus and many membrane-bound organelles. Let's discuss first eukaryotic cell. Eukaryotic cell, from its word eu, which means true, and karyo, which means nucleus. This illustration shows the eukaryotic cell structure. Eukaryotic cells has parts and function. Let's begin with nucleus. Nucleus is the command center of the cell because it is the largest organelle inside the cell. The function of nucleus was to maintain the cell by facilitating transcription and replication process. When we say transcription and replication process, this is the process in DNA. So, nucleus has three components, which is nucleoplasm, chromosome, and nuclear membrane. When we say nucleoplasm, it is based of materials of nucleus, while in chromosome, it is embodied or suspended in the nucleoplasm, and nuclear membrane serve as a skin around the nucleus. Next part of eukaryotic cell are cell membrane. Cell membrane, it is also known as plasma, cytoplasmic, or cellular membrane. The function of cell membrane is to regulate the passage of nutrients, waste product, and secretion in and out of the cell. That is why it encloses the cell. Cell membrane also composed of large molecules of proteins and phospholipids that separates the inside of the cell from the outside world. Cell membrane also has a selective permeability or a property of selective permeable. Next part of eukaryotic cell structure is cytoplasm. Cytoplasm is a semi-fluid gelatinous nutrient matrix. The function of cytoplasm contains storage of granules and variety of organelles, which each organelle has a specific function where the cytoplasm can occur and in metabolic reaction. Next to cytoplasm is endoplasmic reticulum. It is a highly convoluted system of membranes arranged to form a transport network in the cytoplasm. Endoplasmic reticulum has two kinds, which is rough ER or smooth ER. Rough ER has a ribosome attached to it, whereas smooth ER does not. The appearance of rough ER attached to the outer surface of the membrane. Rough ER involved in secretion and membrane protein synthesis, site of glycosylation of proteins, while in smooth ER, it involves the synthesized lipids, phospholipids, and steroids. Is ribosome. Ribosome consists of ribosomal RNA or rRNA and protein. Ribosome plays an important part of synthesis of protein. Next is Golgi complex. Golgi complex connects or communicates with ER. The function of Golgi complex is to complete the transformation of newly synthesized protein and packages them for storage or export. So, Golgi complex the description of Golgi complex is simply stack of flattened and membranous sacs. So next is lysosome and peroxisomes. It is originated in Golgi complex. Lysosome contains lysozyme and other digestive enzyme. Lysosome is found in all eukaryotic cells. It also breaks down a worn out parts of cell or the whole cell, while in peroxisomes are membrane-bound vesicles where hydrogen peroxide or H2O2 is generated and broken down. It is also found in most eukaryotic cells. It also breaks down long-chain fatty acids so they can be utilized by the mitochondria. Next is the mitochondria. Mitochondria is also known as the powerhouse of the cell or energy factory. So, it is where ATP or adenosine triphosphate molecules are produced within mitochondria by cellular respiration. Mitochondria varies depending on the activities required by the cell. One of the activities was when energy is released from glucose and other nutrients. Next to mitochondria is plastids. Plastids are membrane-bound structure containing photosynthetic pigments. There are sites of photosynthesis, so plastids can also occur in light energy to chemical energy. One of the type of plastids are chloroplasts because they contain chlorophyll. Next is cytoskeleton, a system of fibers throughout the cytoplasm. There are three types of fibers, which is microtubule. It is composed of protein subunit called tubulin. Next is microfilament, intermediate filament. It serves to strengthen, support, and stiffen the cell and gives the cell its shape. Next is cell wall. It is an external structure that provides rigidity, shape, and protection. Eukaryotic cell wall contains cellulose, pectin, lignin, chitin, and some mineral salts. So the last part in eukaryotic cell structure are flagella and cilia. When we say flagella, it refers to as the organelles of locomotion, while cilia, it is shorter and thinner and more numerous than flagella. The difference between flagella and cilia: flagella can be found in spermatozoa and certain protozoa, while cilia can be found on some species of protozoa and certain types of cell in our bodies. The similarities of flagella and cilia is they contain microtubules. So the next type of cellular are prokaryotic cells. Prokaryotic cells are classified in bacteria. When we say pro, it means before, while karyot means nucleus. This shows the illustration of prokaryotic cells. So prokaryotic cells have functions and parts. So the first parts of prokaryotic cell are cell membrane. The cell membrane in prokaryotic cell and in eukaryotic cell are the same. So the function of cell membrane is flexible and thin and cannot be seen with compound microscope. Next to cell membrane is chromosome. It consists a single long and supercoiled circular DNA molecules which serves as the control center. Also, chromosome is capable of duplicating itself, guiding cell division, directing cellular activities. Next to plasmid is cytoplasm. Cytoplasm is a semi-liquid consists of water, enzyme, dissolved oxygen, waste product, essential nutrients, proteins, carbohydrates, and lipids. So cytoplasm has a types. First is the cytoplasmic particle. Cytoplasmic particles occurred in cluster and mostly found in ribosome. Cytoplasm is bacterial cell wall. It defines the shape of bacterial cell. It also consists of micromolecular polymer known as peptidoglycan or murin. Peptidoglycan has two types, which is gram-positive and gram-negative. When we say gram-positive, it has a bacteria that have a thick layer of peptidoglycan, while in gram-negative, bacteria have thin layer of peptidoglycan. Also remember that archaea has a cell walls that do not contain peptidoglycan. So this shows the illustration of gram-positive and gram-negative. So the next is glycocalyx. Glycocalyx is a slimy gelatinous material produced by a cell wall, cell membrane, and secreted outside the cell wall. So glycocalyx has two types, the slime layer and the capsule. Slime layer is a not highly organized and loosely connected, while capsule is a highly organized and firmly connected. One of the experimentation that explain the glycocalyx is capsule staining. Next to glycocalyx is flagella. Flagella is a thread-like protein appendage that enable bacteria to move. It also contains thread of proteins which called flagellin. Flagella can be classified into four. First is Petritrichus, Lopotrichus, Amphitrichus, Monotrichus. When we say Peritrichus, it possesses flagella over the entire surface, while Lopotrichus possesses tuft of flagella at one end, and Amphitrichus possesses one or more flagella at each end, and lastly, Monotrichus possesses a single polar flagellum. This shows the illustration about the classification of flagella. Next to flagella is pili, also called as fimbriae. It is a hair-like structure. It is composed of polymerase protein molecules called pilin. So, pili are thinner than flagella because it have a rigid structure and are not associated with motility. They are also enable bacteria to anchor themselves to surface. Some bacteria in pili possess a sex pilus for conjugation. This is the illustration of pili, or also known as fimbriae, and the illustration of flagella. Next to pili is spores, or also known as endospores. It means a survival when there moisture or nutrient supply. It is also capable of growing and dividing. Spores also resistant to heat, cold, drying, and most chemicals. So reproduction of organism and their cells. Reproduction refers to the manner in which an organism reproduces. In cell reproduction, it refers to the process by which individual cell reproduce. Prokaryotic cell reproduce by a process known as binary fission. When we say binary fission, it is the reproduction of prokaryotic cell. It is also the process in which one cell splits into half to become two daughter cells. Always remember that generation, time to time, it takes for binary fission to occur. So this shows the illustration of binary fission. So take a look to the comparison between eukaryotic and prokaryotic cell. This is the end of our discussion. I hope you learned something, and if you have questions, just raise it in our GC. This is Cheri Domingo, your pre-service teacher.